Transcription translation, also known as photosynthesis or gene expression. This is the process of turning code into functional protein. The processes work between two different ways called transcription and translation. Step 1. Transcription. Copying the gene into RNA. This all starts at the process of transcription when a signal, a molecule like glucose or insulin or some other stimulus, is received on a membrane receptor on the outside of the cell. Transcription. A copy of the gene is made from the DNA. This copy is made into a strand of RNA. In the nucleus, the copy is made by RNA polymerase from the template strand. RNA is very similar to DNA, except it is single-stranded and uses a uracil instead of thymine. Right there. Now I'm going to pass it on to my partner.
The DNA will be copied into 3 to 4 directions. The complementary bases are adenine, uracil, and cyclin and glutamine. This is the same as DNA base pairing, but now it binds with U instead of a T, like this. You see this DNA strand? If it was a RNA strand, all the T's would be U's. The copy gene in RNA is called messenger DNA, or mRNA for short. Once the copy is made, it will leave the nucleus and head towards the translation site at the ribosome. Passing it off to my partner now, Travis.
Step 2. Translation. The RNA strand gives information to make proteins. mRNA is read at the ribosome, and the protein is built from the instructions on the mRNA. The mRNA enters the ribosome, which is actually made of 2 parts. The ribosome reads the mRNA in 3 little words called codons. Each codon codes for 1 amino acid, which are the building blocks for proteins. As the mRNA reads each codon, another kind of nRNA, called transfer RNA, brings the amino acid to the ribosome, and the protein is built. After the protein is built, it is either used inside the cell or sent to the Golgi apparatus or the Golgi complex for packaging, and it exits the cell. Now, George will tell you about the protein restaurant analogy.
Now just point to him the whole time.
So, think of all the cells as very extensive restaurant. It's a restaurant, right? So, there's a huge library of recipes, and then the nucleus is the cell of the library, and the recipe books are the DNA. So there's 46 different books representing the 46 chromosomes you inherit 23 from your mother and 23 from your father. Protein restaurant analogy. The process of transcription happens inside the library. So the nucleus and the photocopier RNA polymerase right here, and makes copy of the mRNA of the particular recipe, a gene for a protein. This copy is then sent out to the kitchen, the cytoplasm, so it's the whole cell outside, to the chef, the ribosome, just over here. So, yeah, and then the ribosome reads the copy of the recipe of the mRNA and calls the line cooks tRNAs to bring the different ingredients to the amino acids. The tRNAs always bring the same ingredients. There are actually 20 different ingredients, amino acids, used to make all the dozens of different foods, proteins that a restaurant, the cells, serves needs to survive. And that's it. That's it.